Good morning, everyone, and welcome to the MPG Primer. And today, it is my great pleasure to introduce Marc Elosua Baez. He is a computational biologist in the Cell Discovery Network, an initiative within Boston Children's Hospital, which I've used for help so many times. He aims to use the power of single-cell and spatial resolve technologies to provide precision medicine treatments. Using high-resolution technologies to interrogate biology, he wants to better understand disease-driving mechanisms, where a patient sits within the disease landscape, and how to effectively treat it. From the Cell Discovery Network, he wants to spread single-cell biology knowledge and help more people to analyze their data. So today he's going to be talking about introduction to single-cell RNA-seq workflow. Please feel free to ask questions throughout. There's 2 mics in the middle of the room, and you can also wave your hand and we'll come over to you. Thank you very much.
So I'm going to be talking a little bit about the introduction to single-cell RNA-seq. I don't know how familiar everyone is, so I just want to provide general ideas of how to analyze this data, what considerations, and try to give sort of an intuition of why we're making specific decisions that we believe are very important in the analysis. And these are tool agnostic, so regardless of the tool that you want to use, you can go ahead. And I think we also don't want to focus on any one specific tool because there will be constant tools coming up. So just providing the baseline of when I'm doing this sort of analysis, what should I be looking out for, what are the limitations, and what is happening under the hood at a broad level. So for the agenda, we're going to start a little bit from the basics. So once we have our count matrix or gene expression matrix, how do we go to the graph representation of the data? And then I want to dive deeper into the quality control and the batch effect and integration assessment, which I think are 2 of the key parts of the analysis. And then just very quickly provide some data resources and tool resources that I think could be very useful. But before we start, why would we ever want to use single-cell RNA-seq? And I think I just put a few things here, but first of all, it allows us unprecedented levels of resolution. So it really allows us to understand cellular heterogeneity at a unbiased, quotation marks, way. Since we're going very deep into this biology, it can allow us to also detect novel and rare cell states. Depending on how we design our experiment, it really helps us map lineages and development trajectories, and ultimately can lead into insights into how disease progresses in the mechanisms of it. So basic steps is, I want to start by being where do we want to get to once we have our data? I feel like not many people know this, but single-cell RNA-seq works on a graph structure, and I'm not sure I'm going to try to put too much math in here, but just to get the understanding that we're working on the k-nearest neighbor graph, or we want to get to a point where we can build this k-nearest neighbor graph where each cell is a dot, and these cells are connected between them, regarding on how similar they are between them. How do we get from our count matrix to here is the complicated part, because you can imagine that as we start to have more and more cells, the problem becomes intractable in terms of how you can compute distances between these cells. So let's say we start with our gene matrix, which has 30,000 genes and 100,000 cells. We can't compute distances in this super high dimensional space. And it is neither memory efficient or computationally feasible to really just ask a computer to do this. So we need to find a way to somehow reduce these dimensions. And the way that we do this is we need to first select, or most analysis usually start by selecting a subset of highly variable genes. In order to accurately be able to select these highly variable genes, we need to normalize our data. And some of you might be very familiar, but imagine we have 2 cells, red cell and blue cell. And let's say if we're just looking at the raw counts, we would say, okay, the red cell and the blue cell have the same expression of the pink transcript, and the red cell expresses more of the the teal, and the orange transcript. Obviously, depends on the library size, depends on the sequencing depth, so we need to normalize this. There's many ways of normalizing it, many different algorithms. Normalizing by the library depth usually works super well, and that's what we tend to suggest before trying fancier methods. And once we have the data normalized, we can go on to looking at which are the genes that are most highly variable. This is because we want to subset from our 30,000 gene space. We want to just do a selection of, let's say, which are the most biologically relevant genes. And we define biologically relevant by genes that are variable within our data set. We can imagine that if a gene is constantly expressed, it's not necessarily adding any information to our data set, in the sense that it's not going to allow us to tell us apart different cell populations. So we select our most highly variable genes, and we're going to subset from 30,000 genes. We're now going to look at these 3,000, 2,000, 5,000 genes of interest. Feature selection is very, very important because, as you can imagine, we're going from 30,000 genes to 3,000 genes. Those other 27,000 genes will be invisible. We'll be blind to them for the rest of the analysis. So just keep in mind that the single cell usually is an iterative process where you annotate the level 1, then you subset your data, and then you need to redo these steps, redo the feature selection. Because at each level, you're only working with these 3,000 highly variable genes. Anything that's outside the 3,000 will be invisible to the analysis. And then once we select our highly variable genes, we can scale the data and we can compute PCA. And then we go to, say, 50 principal components that were computed on these 3,000 variable genes for 100,000 cells in our data set. This is very important because these 50 PCs have been computed on these 3,000 highly variable genes. Imagine if we start with a data set that's, let's say, a tissue biopsy that has epithelial cells, that has immune cells, that has stromal cells. The highly variable genes will include genes that come from the epithelial, genes that come from the stroma, and things that come from the immune subset. If then you subset to just your immune cells, you need to reprocess it. So now you recompute these highly variable genes to genes that are highly variable within your immune subset. And that will allow you to gain, let's say, an extra layer of heterogeneity to pull things apart. And I'm just making a lot of emphasis because I feel like a lot of people miss this step, and it's very, very important to really nail this down. And once we have our data in this 50 PC or in this reduced dimension space here, I'm putting principal components. It could be anything. It could be if you compute NMF, if you do SCBI, if you do single value decomposition, if you do any sort of integration method. It's just a reduced dimension space. We can then compute distances, and we can then go on and build our cell connectivity. And this is just showing on PC1 and PC2, but you can imagine that this we're working on 50 principal components, or yeah, we're working in that PC space. Once we have our graph built, our KNN graph, we usually end up with something like this. It's not that we're looking at it, but this is what's happening under the hood. And we want to say, can we cluster this? Classical methods like k-means or hierarchical clustering don't scale well with these super large data sets, especially when we have hundreds of thousands of points. So what we do is we end up using community detection algorithms like Louvain or Leiden more recently. And these basically try to identify communities within our graph that are more interconnected within them than outside of them. So it's trying to optimize the modularity of this graph, but basically it's being like, okay, what cells are more interconnected between them than outside of them? Here I put that SNN, which means shared nearest neighbors. Most of these algorithms don't work directly on the k nearest neighbor graph, and k is whatever number you want to say, 20, 50, 100 nearest neighbors, but more on the shared nearest neighbor. So once you have a cell, for each cell, you look at how many nearest neighbors you share with them, and that kind of prunes extreme points that might not be super related, but just because it cleans up a little bit of the technical variability. With these graph clustering algorithms, we can increase the resolution, and that can further split the initial subsets that we had. And I think one of the questions that we also get a lot is, how do we assess if a clustering resolution is good or bad, or when do we call a clustering done? There's no clear answer. I think biology is ultimately the main driver of this decision, but we can use some metrics. And here I'm talking about two of the metrics that we tend to use. Let's say we have our clustering done, and each column here is one of the clusters. And here we have our samples. We have our flu samples in blue, our normal samples in green, and our COVID samples in red. If we look at clusters 4 and 6, we can see that they're dominated by just one of the patients. Doesn't mean that it's a bad thing. Could be biology, but there's definitely no sample diversity within these clusters. And even cluster 5 seems to be mainly dominated by one of the flu samples. So something to keep an eye out, something that could be pointing that there's something off or different from that one sample, and that it could be worth paying some attention and potentially removing it if it's not going to be useful or we think it's low quality. And we can do this based off the eye test and just drawing the bar plot and being like, what does this look like? Or, for example, we can compute the Simpson diversity index for each cluster, and we can see how diverse is each cluster by the sample ID or the batch that we're looking at.
I have a quick question.
Yeah.
So if you get a cluster like 4, how do you go about deciding if that's some weird batch effect or if it is some unique biology that's like, do I kick it out or investigate it?
That's the question. That's why I said at the beginning that we can provide technical expertise, but knowing the biology is the most important part when making these decisions. In this case, I believe cluster 4 ended up being platelets. So it was a sample that just had a lot that wasn't cleaned up well in terms of the blood wasn't fully removed. So we had a lot of hemoglobin genes in cluster 6 and a lot of platelets in cluster 4. So we ended up removing these because they were not a cell type that we're directly interested in, and that had just missed let's say, the sample processing quality steps. But again, just by looking at this, I would not remove the sample, that cluster. I would interrogate it and be like, what genes are differentially expressed? Do I see this in my condition of interest? Because if I see this cluster just in one of the patients, but it's in, let's say, in my disease category, then I might be more inclined in keeping it to be like, oh, this seems to be something that's more disease specific. But I'm going to go over some QC metrics in a little bit as well to try to help with that process. But again, I think biology, if there's one point, is that biology is the key driver of the decision making. We can provide as many parameters and tools and things to look at, but laying our decision making on the biology is I think the ultimate driver of what's going to make a successful analysis. And another thing that we can do is we compute the silhouette score, which the silhouette score basically means is my cell more similar to the cells in its own cluster than to the cells outside of my own cluster. And I'll show the formula in a few slides, but basically what we can see is that high silhouette scores near 1 means that the cells within that cluster or that cell is very similar to self than to other. And scores 0 or below means that cell is equally similar or more dissimilar to self than to others. So we can see how, let's say, clusters 8, 7, 6, 4, 3, 2, and 0 seem to be very well structured, seem to be very similar to self. Then all the cells within those clusters are similar to the cells that are also in the cluster than the rest. Cluster 5, for example, which is, in this case, is this one over here, seems to not be doing such a good job. So cluster 5 could be something potentially interesting to look at because the cells within cluster 5 seem to be more different between them and more similar to stuff outside of the cluster. Cluster 5, when we ended up looking at it, were a lot of proliferating cells. So they got grouped together because they shared the proliferation signature, but there was a lot of cellular heterogeneity within them. So when you were drawing distances, The proliferating T cells were still similar to the T cells. The proliferating myeloid cells were more similar to the myeloid cells, et cetera. So it's just an indication. Doesn't mean that it's a good thing or a bad thing. It just means that it's something that we need to take into consideration and look deeper into before we make a final decision. So once we have the clustering, based on this k-nearest neighbor graph, we can also compute our UMAP visualization of the data, which I'm sure most of you have seen some sort of UMAP by now. I just want to caution that you can see that we started from 30,000 genes at the beginning. We subset it to 3,000 genes, and then we reduced that to 50 principal components. We built a graph on that, and then we reduced this to 2 dimensions. So it's been a long process, and a lot of data has been, let's say, shed or compacted during these steps, because we ultimately want to see, can we capture the most amount of variability within 2 components? This doesn't mean that there are more sources of variability beyond these 2 components. And the distances that we see within our UMAP, don't overinterpret them. Just because you see the 2 things are very close together and one thing is very far apart doesn't mean that that's necessarily biologically true. It's just a way of visualizing the data. If we added a 3rd UMAP dimension, maybe the green cluster would be closer to blue than the orange, just because with the 3rd dimensions, NK cells would go all the way up. So just to not overinterpret UMAPs, there can be useful to look at the data and just have some visual reference, but yeah, don't judge distances and relationships. And if you see 4 clusters right in a row, don't assume that this is one transitioning space that's going from one to the other, because basically you're just drawing things to be like, you're compressing 30,000 genes into 2 dimensions. There might be a lot that's missing there as well. And once we have this, then we can go on to do our downstream analysis. We can do differential gene expression. We can annotate our cell types. We can do some sort of compositional analysis. We can do cell-cell communication, many downstream analysis. That I think that really depends on the question that you're trying to answer in your data set. For the quality control, I want to spend some time because I feel like it's a very important part of the process, and some people sometimes rush through it. I tend to be way more conservative in the way that I do quality control. I will only remove something if it's very clearly obvious that it's poor quality, or let's say an empty droplet. I will start with the sample level metrics. So once you've done your sequencing, can we look at the metrics that you obtain from sequencing your data and then mapping them to your reference genome and see what we're obtaining from them. Here I'm putting on the left the, let's say, typical cell ranger report that you get when you've done a 10x genomics experiment and you run it through cell ranger. These are the basic summary statistics. And basically, by looking at these sample level metrics, we're just trying to understand, was this sample good quality? Was there something off? Are we getting the expected number of cells that we were expecting? Is the quality of the sequencing good? The sequencing depth, is it the one that we expected from the sequence facility? Did mapping these cells or these reads to the reference genome work well? Did it not? And try to get ahead of some of these QC analysis or QC problems that we might see later on downstream, but try to identify them earlier on at the sample level and be like, okay, we know that anything that happens within the sample could be biased because it wasn't sequenced properly or it wasn't sequenced at a lower depth, et cetera, et cetera. And this, I like to look at it in 2 main different ways. So one is on the sequencing and alignment quality. So we're looking at the sequencing depth, which hopefully you can read, but we can get the total number of reads that were sequenced, how many of these reads, or how many of the barcodes and UMIs that were sequenced are valid, and UMIs are unique molecular identifiers. So when you're doing your single cell experiment, each individual transcript will be assigned a unique barcode. So when you do the PCR amplification, et cetera, you can correct for amplification biases. Just UMI is short for this unique molecular identifier. And then we also have the Q30 scores to see how good the sequencing was in the barcode, in the RNA read, and the UMI. These usually always look very good, but sometimes you'll see that some specific samples just look very, very poor, and that could be like already an indication that that sample could be troublesome in downstream analysis or might not be worth adding in here. And then some other things that are interesting is percentage of reads that were mapped to the genome, and then reads that were mapped confluent to the genome, mapped to intergenic, exonic, or intronic regions. I can't give you a number of what number you should be looking for here. It will vary. If you're doing single-cell RNA sequencing, you will get a higher proportion of reads mapping to the exonic regions. If you're doing single nuclei, where you're really just looking at the stuff that's within the nuclei, then you will get a higher percentage of reads mapping to intronic regions for the more immature RNAs. So just to keep in mind, how did the sequencing and how did the mapping of our reads go? And then we can also go to the sample and cell quality. So usually people tend to load 10 to 20,000 cells. So are we getting the expected number of cells? Sometimes you get 3,000, sometimes you get 50,000. If you get 50,000, you could be like, did I overload it? Did something go wrong and it's considering more cells than it should be considering? We have our barcode rank plot up here that I will talk in a second. But yeah, this basically determines the estimated number of cells, number of reads that ended up in a cell, some statistics on like how many molecules, how many genes were detected per cell, et cetera. And I really like to look at the sequencing saturation and at the barcode rank plot. The sequencing saturation is this top plot where we have the mean reads per cell and the sequencing saturation in the y-axis, meaning that as we sequence deeper and deeper, the likelihood that we're going to find new genes gets lower and lower. So at some point, there's this trade-off, right? Around here, let's say, where in order for you to get a more, let's say, complex library, more new information, you need to sequence way deeper. And in most of these cases, it's usually better to sequence more samples than to sequence deeper one sample, because you usually end up having enough information by 0.6, 0.7. And then the barcode rank plot is the automated way that Cell Ranger has to determine what is a cell and what is not a cell, or what is an empty droplet versus what is a droplet that has captured a cell. And you can see that here's the number of barcodes in the x-axis. Here's the number of UMI counts per cell. All the cells that have many UMI counts end up being considered a cell, and then it usually gets to an elbow point. And right after the elbow point, it will set a threshold, and it will be like anything after this is not considered to be an empty droplet. Everything above this is considered to be a droplet with enough information to be considered a cell. This can be misleading. Always, I like to take a look at it because sometimes you get a double elbow. If you have granulocytes, for example, that have very low transcript mRNAs, you can get this double elbow, and by setting this automatic threshold, it could absolutely just remove a whole population from your analysis. So if you're expecting to see something, you don't see it. It could be because in this first step, and this is when you run Cell Ranger, you get your filtered gene expression matrix, and you'll get your raw gene expression matrix. Your filtered gene expression matrix will have around 10,000 cells. Your raw will have all of them, so we'll have 1 million cells. It's much easier to just start from the, let's say, pre-filtered one. But if you see, or if you have the intuition that something's off, that you're missing something, you can always access all of the data that's in this tail end over here, and really try to recover as much as possible. Moving on to the cell level matrix, here we just want to mainly remove empty droplets, so droplets that have no genetic material within them or just have ambient RNA. We want to identify doublets and at least label them as potential doublets. And we want to remove noisy artifacts. Some of the key metrics that we look at are the library size and the library complexity. Library size is how many UMIs we detected for that droplet. Library complexity is how many genes we detected for that droplet. Mitochondrial percentage is what percent of genes map to mitochondrial genes, and the doublet score we'll look into a second. But we can imagine that this is all our droplets after running the microfluidics section, and we see that we have some empty droplets. We see that we have a cell that's stressed out and that's dying. We see that we have 2 cells within 1 droplet, and we see some empty droplets that have some ambient RNA. I'm not going to go into ambient RNA removal because it's a little bit more advanced and don't necessarily recommend to do right off the bat, but it's a thing that could be happening within your data set. And if there's just like, sometimes you just find underlying baseline expression of a gene throughout all of your cells. It could be ambient RNA worth removing. But some of the metrics that we're using are library size and library complexity. If we see cells that have very low library size and very low complexity, most likely they're going to be these empty droplets. We can label them and start removing them from our data set. We can look at our mitochondrial percentage. It usually identifies dying or stressed cells. Usually when cells get stressed, they'll like have some pores in the membrane. All the cytosolic content or mRNA will leak out of the cell, but the mitochondria, which are too big, will remain enclosed. So when you sequence them, you'll see very, very high mitochondrial percentages within those cells. And again, very different if you're doing single cell versus single nuclei. If you're doing single cell, you can get some cell types up to 30, 40, 50% of mitochondrial percentage. I think I have an example later, but like cardiomyocytes, which are muscle cells, will have very high mitochondrial percentages, up to 40, 50%. That doesn't mean they're low quality cells. It's biologically driven. So you shouldn't remove a cell just because it has a high mitochondrial percentage. But if you're doing single nuclear RNA-seq, where you're just working with the nuclei, you really expect to find very, very clean cells where there's no mitochondrial percentage.
Question related to single cell versus single nuke. We do single nuke because our cells are often in syncytia, so we need the separate nuclei. But then we really have to clean up the ambient RNA, I think, because the cytosolic RNA sticks to all the nuclei. Is that just us, or is that something you see generally?
No, I think with the single nuclei, you can get more ambient RNA just because during the process it may not be fully clean or might be sticking.
And you use cell bender in those cases?
Yeah. I think cell bender is one of the most used tools for it. And again, it's one of those tools that works very well, but it really requires a lot of control, right? Because like if you use it, if you're too strict with it, you might start to remove true biology. So always better to start a little bit more conservative and then bring it up more and more strict as you go. And with the doublet scores, we want to identify instances where 2 cells ended up in 1 droplet, and we can't confidently say that it's A or B because it's A and B together. Want to make an emphasis into doublet dissection algorithms. There's many. I put 4 down here in the bottom left corner. But just to like get a sense of how these algorithms are working. Basically, what they're trying to do is they're trying to mimic a doublet that has happened during the process. And to do that, you start with, let's say, you have your PC space with all of your cells. And what you do is you create artificial doublets. So you grab 2 random cells from your data set, you put them together, and you do that many, many times. And you put those cells back into, or these simulated doublets back into your reduced dimensions embedding. And we can see how, in some cases, all of these simulated doublets end up clustering together, meaning that these droplets that we were seeing here are most likely technical doublets because they're surrounded by artificial doublets that we generated. But sometimes it's not as clear cut. Sometimes doublets look like a mix of 2 phenotypes. Sometimes if you have a cell that's differentiating from point A to point B and it has a phenotype that's A and B like, you can find that these cells get very high doublet scores, where that's just biology, it's because that cell's transitioning. Sometimes we see this a lot with intermediate monocytes, where they end up usually having very high doublet scores because they're this mix of classical and non-classical monocytes where they're expressing CD14 and CD16. So just because a cell has a very high doublet score doesn't mean that it's a doublet. It could be biologically relevant. Again, it's just a moment to make an informed decision on your biology of the data to be like, do I expect to see this? Do I not expect to see this? Could this be biologically relevant or not? Obviously, if you see T cell and B cell marker genes within one droplet, that is very indicative that is most likely a doublet. But in these like intermediate cases where it looks kind of fuzzy and kind of transitioning, be careful before removing something, because once you remove something, those cells are gone. You're not going to remember that you removed them. That's gone. That's out of the picture. So always better to be a little bit more conservative with what you keep than with what you're removing. And when we're looking at these cell level metrics, I mentioned them before one by one just to like see how they could help us. Just looking at them one at a time is not enough. We need to see how these things vary together. So for example, we can look at the number of counts, so the library depth, the number of transcripts detected for each cell, and the library complexity, the number of genes detected. And we can see that as we increase the number of counts, we increase the number of transcripts detected. But sometimes, for example, we see that if we look at the hemoglobin percentage, it's very high down here. So if we were to remove all of the cells that have less than this number of counts and that less than this number of features, we would be removing all of our red blood cells. We're usually not interested in red blood cells, but if that was your population of interest and you just set hard thresholds, say bye-bye to your data because you just filtered it out of your analysis just by setting hard thresholds. Some of the things that you also see sometimes are these tails down here, where the library complexity goes up, but the number of features doesn't necessarily go up. And here the dots are called by the mitochondrial percentage. Most likely it's low quality cell, so I'm going to show an example of when it's not a low quality cell and why we need to be careful with this. And then I also like to, in this same plot the scrolled doublet scores, because one of the things that we could think is these cells that end up in the top right corner could be doublets because they have double the genetic material. So we expect slightly higher number of counts and number of features detected within there. But if we look at an example here from everything that I just told you, we would be like clusters 2, 4, and 6 potentially, right, seem to have very low library size, very low number of detected genes compared to the rest. And again, clusters 2 and 6 seem to have low mitochondrial percentage and low ribosomal percentage. So what could be going on with these clusters? Are they low quality? And most of these clusters ended up being in this lower tail down here. But before we removed them, we just did like a standard differential expression analysis between our clusters. And what we saw was that cluster 6 expressed LY6G and some CXCR2, and cluster 2 expressed a lot of CXCR2, as well as IL-10 and arginase-2, meaning that for us, these were very clearly neutrophils that were included in our analysis. And if we had set a big threshold, these neutrophils would have been completely removed. And again, if you look at cluster 2, it has a big expression of IL-10B, arginase, and CXCR2 compared to the rest of the data set. If we go back one slide, cluster 2 seems flat as a pancake in terms of the library depth. So just because something has a low number of features or low library complexity, don't just remove it automatically. Take a second to look at what are the genes that are differentially expressed before kicking it out of your analysis, because you could be missing a population that just happens to have those characteristics. And the same thing with cardiomyocytes. They can have very high mitochondrial percentages. If you just set a threshold of everything that's over 30% mitochondrial percentage, I'm going to remove, you could be missing out on a complete whole population that just biologically happens to have higher mitochondrial percentage. And the last thing that I want to do is, I don't like to remove cells with these strict thresholds. So what I tend to do is, let's say we have our all of our cells. I will label my cells as good quality and bad quality depending on fixed thresholds. But before going on and removing, I will do a differential expression between the good quality cells and the bad quality cells, and just see if any biologically relevant genes are popping up to be overexpressed in the cells that I labeled low quality. And if they are, then I will keep those cells in. And I will in iterative rounds of the analysis, I will be maybe a little bit more, I will find a little bit more noise and technical noise in there, but I'm making sure that whatever population was labeled as poor quality is kept within my analysis. So I really like doing this. I think it's called diagnosing cell type loss. I think it's super important. It can really help keep all the biology that you have in your data set and avoid you removing things. So that's for the QC part. Moving on to batch effects and integration assessment. Batch effects will always be there. Batch effects happen at any point of the process, different location site, different sequencing machine, different person processing the data. They will happen. It's just something that we have to deal with. The question is, how do we go about it? Most important thing is experimental design. If you have a good experimental design, it's so much easier to correct your batch effect. So let's say we have two experimental designs. This top one, where we have, let's say we have control, disease 1, this is disease 2, and we sequence each one in its own separate batch. And then we see differences between them. We don't know if these differences are due because of the batch or because of the underlying biology. We can correct by batch, which will tend to homogenize our data set, but that means that we're also correcting for biology at that step, because we don't have any way to control biology and batch separately. In our second experimental design, where let's say we grab our control, disease 1, disease 2, and then in our batches, we mix control and disease 2, control and disease 1, and disease 1 and disease 2 into 3 batches. Then this allows us very much to correct things that are happening. So we'll see, for example, these box plots are colored by batch. Here we have a very clear batch effect, right? Where all the samples that were sequenced in batch 1 have the same. So we can say, okay, proportion of detected genes in this case seems to be driven by batch. This is something that we should be taking into account. Something that can be fixed computationally, but it's so much easier when done already from the experimental design side. And sometimes when we're fixing it experimentally, what we're actually doing is we're just killing very important biological signal just because we were forced to correct these things. Or if we don't correct them, then the results that we're seeing, we don't know how much of them are true biology versus just technical noise. Sometimes batch with single cell looks like this. This is just looking at it on the UMAP, where you see how these populations seem to be very similar, but they're just mirroring each other. So how do we get them to integrate much better? And post-integration, we want to obtain something that looks more like this, where both conditions are much more overlapping. I just want to go about just let's not just look at a UMAP and be happy with how it looks and assess integration that way, but provide some metrics that can help us assess how the integration has gone and assess it. There's a very there's a lot of nice benchmarking papers. I really suggest a Malta-Lucan paper. I think it's from 2021 about the integration benchmark, SCIB they call it, because they really are very thorough in how they go through integration. But I want to talk about these 3 metrics, the I-LISI and the C-LISI score, the silhouette score, and the PCA regression. There are 3 metrics that we can use to assess how much or how well the integration has worked. If you look at the first one, the I-LISI and the C-LISI, they're the same thing, but the name changes because what we're looking at here is batch on the left versus cell type on the right. So if you can imagine where we have a perfectly annotated data set and we integrate it, we would want batch to be completely integrated, while cell type labels remain distinct between them. So all the cells that fall within the same cluster should be from the same cell type label and very heterogeneous for batch. There's the local inverse Simpson's index. We can compute it from the batch. So if we computed it with the batch variable, we want this variable to be very diverse. We want it to be very high. We want to find very heterogeneous neighborhoods in terms of batch within each cluster or within each cell. And we also want to find this C-LISI or the cell type LISI very low. We want this cell type heterogeneity to be very low. Since we usually don't have the predicted cell type right off the bat, what we try to do is some sort of automated cell type annotation that gives us broad labeling of our cells and try to assess it that way. So the way that we're trying to assess the integration is we want to increase the iLISI, increase the LISI score over the batch covariates, and decrease it or keep it as much as possible for cell type, for the cell type label. So we keep our distinct cell type neighborhoods while we merge them across batch. We can also compute the silhouette score that I mentioned before, which is basically the distance of each cell versus the centroid of its cluster versus the distance of that cell towards the centroid of the nearest cluster in our data set. If we have clean data, and here we're looking, let's say, at the batch covariate, we would not want to find two distinct clusters if we're assessing a batch covariate because we would want these to be very merged. We will want to correct that effect. So if we go back here, this would be an example where this silhouette score would be very high by batch because each batch is very similar to itself and very different from the rest. But in these cases, it would be great. Looking at the silhouette width by batch, we want to have high diversity by batch. Look at the silhouette width by the predicted cell type label. We want to have very low because we want to still keep the structure of our biological data together. And then we can also look at the PCA regression. So we know that this is a little bit more complicated, but we know that each principal component explains X% of the variability within our data set. And then we can draw some kind of model where how much each of our batch covariates explains each principal component. And then we try to like align these things, and we look at it before integration and after integration, and we want to see if, let's say, the site of collection was initially explaining a lot of the variance within PC1, but after correcting or integrating, this variance has been decreased. So we've effectively removed the effect of site of collection on principal component 1. Always look at positive and negative controls. So always have things that you know that you want to correct for. So the sequencing flow cell, the site of collection, date of collection, person that handled the samples, versus things that you know you do not want to correct for, or bioconservation metrics. So if you have the predicted cell type label, or you have the cell cycle score, or you have a biological covariate of interest, disease versus healthy, and if it was a well-designed study, you would expect disease versus healthy to not be corrected because that is your key question and you don't want to confound that biology with batch. So again, I think it's very easy for computationalists to just run analysis, whereas wet lab biologists really have like the positive controls and negative controls and everything. And I think we should start doing more of these in our tools. And like when you're running any new tool, you always need to expect something that you expect to find, something that you don't expect to find, and that helps you or run in a case in different scenarios that you can validate that it works, it doesn't work in your hands. Moving on to closing up the stages, possible analysis that you can do with single cell. You can do differential expression analysis between conditions. Usually, people will recommend to do this at the pseudobulk level. So let's say you want to find differences between CD4 T cells in disease versus healthy, you would do pseudobulk by patient, by cell type, and compare them with classic bulk RNA-seq differential expression tools, which are much more robust. And really, once you do this pseudobulk, you reduce the sparsity a lot more. You can extract functional gene programs. You can map gene signatures of interest, identify gene regulatory networks. This works especially well if you add other layers like single-cell attack sequencing. You can do cell-cell communication, compositional analysis, lineage tracing, single-cell QTLs if you have genotyping information, many different things. There's many papers out there. Tools to analyze the data in R, Seurat, and Bioconductor are the two big players. In Python, it's ScanPy, which is part of the scBurs family, which is trying to do this Bioconductor family of suite of packages for Python, which I think is very cool. And if you don't have access to your own data, there's very large data repositories, such as the Cell by Gene database data portal, which has 107 million cells over 1,700 data sets. The single cell portal from the Broad, again, 55 million cells from 800 studies. And the Human Cell Atlas data portal, which only has healthy cells and has 63 million cells from over 9,000 donors. Here, I think that this can be shared. So I put some resources on places to get started or to go more in depth into what I just explained. But these, I think, are three very good resources to get started and revisit to just say the foundations of the analysis. And yeah, I would say that the take-home messages from the talk would be that the pre-processing steps matter, that they can have a big influence. So the way that we go from gene expression to our graph structure and the feature selection, the dimensionality reductions are really key into what biology we're going to see afterwards. The quality control should be carried out cautiously. We can't just remove cells out of a fixed threshold because you could have biology that you're missing out. So please do some diagnosing cell type loss analysis to retain, let's say, weirdly behaving cells that you might not expect. And that batch effects can be corrected with integration methods, but try to have some positive and negative controls of what you're correcting. And if you don't have a good experimental design and you have to end up overcorrecting your analysis because your biological covariate of interest is confounded with a technical artifact, then you will be homogenizing all of that together and losing on a lot of biology. So if you have anyone that you can go talk to, or even you can come talk to us at BCH, we can help in these early stages of just thinking about the analysis, because sometimes we find that people have already gone all the way down the analysis and generate the data, and now we're just trying to minimize the damage. So yeah, I would like to thank everyone in the Ordovás Montañez lab at BCH and also the Cell Discovery Network group and all our funding partners. Thank you very much.
Do we have any quick questions in the room? It's 9:20, but I do have one quick question. With the QC, do you recommend doing like looser thresholds first and then clustering and then doing QC like by each cluster, like iteratively?
Exactly.
So you don't remove the weird cells?
Yeah. So the way that I end up doing most of the analysis is I'll label them as good, bad quality. And then you'll go through level 1, level 2, level 3 integration. And at each level, I will keep all the cells, unless there's like cells that are like really depleted of transcripts. But at each level, you will identify clusters that seem to be behaving weirdly, and then you can contextualize them within that level. So I'd say once you get within CD4 T cells, you see a weird cluster in there, and you're like, okay, these seem to be, this cluster seems to be enriched in lower quality cells, and then you can remove it there. Or it could be, biologically relevant, and you might want to keep it. Mm-hmm.
It does seem easier to label them as bad and remove them later than to go back and start over to get them back. Oh, it's snowing. Okay, that was great. It's just a reminder that this is a science, but also an art, a very challenging one that requires computational skills, but also cell biology
I
knowledge.
think that cell biology is the hardest
Yeah.
part to bring into here.
You almost need like a computational biologist and a cell biologist at this together. Okay. Thank you. We'll see you here next week.